Good afternoon, everyone, and welcome to this week's installment of the ABRF Sequencing Seminar Series. We're delighted to welcome our colleagues at MGI. But before we get started, I want to share a couple of updates on upcoming activities for ABRF. Just let me set this properly. Hopefully you can see that. Of course, we'll start with the big news, which is our annual meeting next year will be May 7 to 10 in Boston. Registration for that meeting will be available by the end of October. For those of you who may be with sponsors, we're accepting sponsor registrations very shortly, and you'll be contacted to provide with more information about that. So I'm going to go back to the top. And again, thank you for being here. We're going to use chat for any questions. Feel free to introduce yourself in the chat, where you are, your institution, where you are throughout the country or internationally. And if you have questions for our presenters, and we hope you do, please post them in chat. That way everyone can see them, because one person's question often leads to another. As you may have heard, today's session is being recorded, and we will share the recording with all of the participants, all of the registrants, and the recordings will also be posted to the ABRF website, as they have been for the preceding sessions as well. Just a couple of reminders of upcoming events. This series continues pretty much on a weekly basis. You can see the other organizations that'll be presenting. Each seminar will be at the same time on Thursdays, 2 to 3 p.m. Eastern. And there's the registration link that you can use to sign up for any of the future programs. ABRF has 2 chapter events coming up next month. The NeuralSkid or Northeast chapter meeting is October 12th to 14th in Rochester, New York. You can see the QR code there. Registration is still open and travel awards are still available. And then our Midwest chapter will be meeting just after that, October 17 and 18 in Columbus, Ohio, near the campus of Ohio State University. And their theme will be Better Together, Strengthening Communities of Practice and Science in Cores. And again, you can follow that QR code to the registration link. And I mentioned the annual meeting. So with that, I'm going to stop sharing and I'm going to turn it over to our friends at MGI. Wenxi, would you like to get us started?
Yep. So let me share my. Can you guys see my screen? Yeah. Hi, everyone. Thank you for coming to our MGI webinar. Today, I'm going to present you the DNB-seq technology and platforms. My name is Wenxi Liao. I'm the Associate Director of Product Management at MGI Americas. So first of all, what about MGI? MGI is a global company. Our headquarters is in Shenzhen. We have 9 international subsidiaries. As of December 31st, 2021, we have more than 2,000 employees around the world. Right now, this year, probably more than 2,800. We have sold 2,700 sequencers in 80 countries and regions. We are really committed to developing technology for life science. And as you can see, we have a very strong R&D team. 1/3 of our employees are in R&D. We also have proven track record. We have more than 8,400 publications, and I will share with you that more later. Our Americas is based in San Jose. It was previously Complete Genomics acquired by MGI. MGI just went public not too long ago at Shanghai Stock Exchange. We offer 3 major product lines. The first one is a genetic sequencers. As you can see, we have a different throughput sequencers. In the middle is a laboratory automation systems. We also offer sample prep or library prep automation system. On the right side is a new products that can be ultrasound system or bioinformatics products or mobile library platform. As you can see, we offer a very diverse portfolio. This is our publication. As August 22nd, we have more than 8,400 publications. Just 2022, we have more than 3,000 publications. And if you look at the right lower corner, you can see the applications, the publications by applications. The most popular one. The most is 1100 applications, 1100 publications on RNA-seq, and followed by WGS and targeted seq. That's actually pretty in line with most popular applications as well. We move on to the next slide. So that's about MGI. Now I'm going to share with you about the DNB-seq technology and sequencing chemistry. So the DNB-seq technology involved from prepare library all the way to make DNB and load DNB. As you can see from this graphic from library, the library is actually single-strand circular DNA, and you go through the rolling circle amplification we call RCA, and it become DNB we call DNA nanoballs. Abbreviation is DNB, and then it will be loaded on the patterned array. So this is our library preparation. Is actually very similar to other library. The only difference is you can see on this, we add this phosphate on the 5' end and plus the splint oligo that actually help this linear single strain DNA to become a single-stranded circular DNA, and that's here. So the splint oligo plus that phosphate make it happen. Next step is to make DNB. DNB is actually from, as you can see, it's probably easier to see this animation on the right side. So from single-stranded circular DNA to DNB, and that's through the rolling cycle amplification. And it's always making copies from the original single-strand circular DNA. Each DNB is the same size with around 300 to 500 copies, depending on the fragment size. But because of this technology, it offers spatial accuracy and lower cost. First, because it doesn't have clonal error, it doesn't have index hopping, and the SNP and indel precision and sensitivity is very high, more than 99%. So our the DNB loading is based on electrostatic interaction, and that to ensure 1 nanoball per spot, and obviously combined with this pattern array. So it's really making sure each ball it will be fit into 1 spot, and there's no preferential loading of small fragments because each DNB nanoball is about the same size, and so the duplication rate can be as low as around 2%, and that low duplication rate equals less sequencing reagents needed. Now I'm going to move on to our DNB-seq sequencing platforms. So now we have just launched our G400 in September for the first time in the United States. However, like I mentioned in the beginning, we actually already have 27 sequencers around the world. So you can see we offer from very low throughput all the way to extra high throughput. So the G400 is our flagship machine. It was launched 2017, and then we have a T7, and that equals to NovaSeq, and that's launched 2018. And then we have T10, and that's super high throughput that was launched 2020. For the US, this one is already available now. For T7 and T10, that's going to be available to ship starting January 1st, 2023. We do have 2 E25 and G99. Those are the low medium throughput, and that's coming soon in 2023 in the United States. I'm going to focus on G400 because that's what's available now, and that's the one and only sequencer that offers the most diversified options. We offer 2 types of flow cell. One is FCS, meaning the S means small, and that has 550 million effective reads, and we have single end pair end options and the read length up to PE 300. We also offer FCL, that's flow cell large, and that effective reads is 1500 to 1800 million reads. And we also have different single end pair end. So you can see we have pretty good wide range of the read length from 50 to 400. The right side is the interior of the instrument. This slide might show you better. So you can see there's number one is a laser on the top, and you can see a camera. The number two, that's a high resolution camera over there. You can see the number three, that's an injection pump, and the flow cell is here. It takes 1 or 2 flow cells. On the right-hand side, there is a control computer inside on the where the 5 is, and then outside the exterior, there's a touch screen monitor, and we have a reagent container and the compartment and the door. There are 2 loading options. The first one is manually loading on the sequencer. So that would require 50 microliter per lane. And this one, you load one sample, one mixture to all 4 lanes together. Second option, the option B, is portable DNB loader. We provide 2 free loaders per instrument. And this one requires less, is 25 microliter per lane. It actually can load 4 different types of mixtures into 4 lanes, because this picture may not show well, but each lane, in front of each lane, it has a hole, and that's where you can load the sample. We offer different applications from NIPT, small RNA, single cell RNA-seq, all the way to transcriptome, WES, and WGS. And based on the read length, based on this is the number of samples for recommended per run, and each run require you can have 2 flow cell. Right now, this is 2 options we recommended for our bioinformatics workflow. So basically, the FASTQ will be generated on the sequencer. And once it's generated, there are 2 options. You can either transfer to your server or cloud, or we also offer the MegaBolt or ZBolt, and that can perform WGS and WES analysis. And eventually, if you like, you can transfer your data to your server. I think we are the one company on the market which actually offer the complete workflow from sample to result. We have as I mentioned in the beginning, that we actually have lab automation systems. So this MGI SP960 can do library prep plus make DNB. And then you just need a sequencer, and then you can have the MegaBolt or ZBolt for your data analysis. We also offer MGI Z-LIMS Lite, and so for the complete workflow. We actually got asked a lot that how do you run third-party library on DNB-seq platforms? So this is a target library. If you are doing the TruSeq or Nestera, it's actually very easy. The first step, you do the library conversion, because our library is single-stranded circular DNA. And so this conversion kit is actually to convert the linear double-strand DNA library into single-stranded circular DNA. And on the right side, it's actually very similar to what I have shown in the beginning. So basically, this PCR primer with a phosphate the primer with a phosphate will make sure that this DNA strand has that phosphate, and then plus the splint oligo, and that's how it convert to single-stranded circular DNA. Next step is to pick appropriate sequencing primers per your library. We have different types of the primer kit. For example, the App A primer kit is for TruSeq library. For the ABC is actually for TrueSeq plus Nestera. And then the last one, ABD, is TrueSeq plus Nestera and MGI. Those two are the most popular options. So because that's easier for the customers to have ABC because it covers both libraries. For ABD, it's really for customers thinking that, okay, now I have TrueSeq Nestera, but maybe I still have space. I can run more libraries, and they combine with our MGI library, and that's what you can choose ABD. After that, you just need to pick the right MGI sequencing reagent set, then you are ready to run sequencing. Please come to visit our demo lab in San Jose, California. It's very spacious. It's more than 800 square footage, and we have our different type of automation equipment and sequencing platforms. Just want to let everyone know our DMB-Seq G400C is ready to ship now. The site, Complete Genomics in San Jose, it was acquired by MGI, is the supply center of Americas. So we supply to Canada, Latin America, and the US, and you will already see is fully stocked with our instruments and reagents. Okay, that ends my presentation. Thank you very much. I'm going to hand over to Yuan.
Thanks, Wenqi. I'm trying to share my screen. Let me see. Let me know if you can see my screen.
Yeah.
Yeah. All right. Hi, everyone. Thanks for attending this webinar. I'm Yuan, VP of Research. So today I would like to give you a little bit background of our technology in the spatial genomics space and some of the ways we've been applying it, and also its specific and unique advantages compared to existing tools in the spatial genomics field. So we believe spatial genomics is a next life science revolution after microscopy and NGS. The breakthrough of spatiotemporal omics technology makes it possible to understand the complex regulation process of cell fate, human cognition of biologic laws behind individual development, disease occurrence and evolution will usher in profound changes. It was selected as a method of the year 2020 by Nature method, and the bulk analysis, we know everybody has been using RNA-seq to look at the bulk analysis, the average gene expression from mRNA in the tissue cells, and single cell RNA-seq was developed. It brought a more fine-grained assessment of each cell's transcriptome. With spatial resolved transcriptomic methods, scientists can get transcriptomic data and how the positional context of those cells in the tissue. And the problem we set up to solve for spatial genomics is that the existing technology still have some limitations, which include the the resolution and capture area as well as capture efficiency. For example, for the ones that have relatively large scales, traditionally is still relatively limited in the amount of information it can capture in space. For example, Visium has a field of view of 55 micron, and and has a field of view of 6.5 millimeter by 6.5 millimeter, which makes it difficult to look at some of the large tissues or organs, for example, macaque brain or human brain at a meaningful scale for the way it's spatially organized. And the second challenge has been resolution, and which Visium offers 55 micron of spot size and center to center distance. And this is two metrics we usually use to measure the resolution. One is the spot size where the probes are located, and then the distance between the capture area. And with visions 55 micron, and with the average cell size being 10 microns, it is still quite far from achieving single cell resolution, let alone subcellular resolution. And some existing technologies like merfish or sickfish have been able to have very high resolution, but also unfortunately limited by its field of view and as well as the gene numbers it can capture and work with. And also it requires a lot of prior knowledge of what you are looking for. And for our method, we are in situ unbiased to capture the whole transcriptomics, and we can achieve more than 500 genes per single cell in a mouse brain, which is the most suitable tissue type for all the spatial transcription tools. And on our 1 centimeter by 1 centimeter capture chip, we can generate more than 1 billion reads. So this is, and we can customize the chip size. From this is what I just mentioned, the 1 centimeter by 1 centimeter, and we can go up to 13 centimeter by 13 centimeter. Here, the largest example is close to 7 times 7, and we have been. For example, for 3.5 centimeter by 5 centimeter, we have been using hundreds of these chips to do a 3D reconstruction of a macaque brain. And this can all be customized. So basically, our technology can give you best of everything over here. We can give you a field of view up to 13 by 13 centimeter, and we are able to get down to a resolution which is truly subcellular of center to center distance be 500 micron and spot size of 200 nanometers. I'm sorry, I forgot to mention here, this is our matrix. This is our spot size is 200 nanometers with cell size being 100 micron. We can have up to 400 capture spots within a single cell. And our center to center distance is 500 nanometer, which is incredible. So in summary, our technology provides a subcellular resolution of 500 nanometer and goes up to 13 by 13 centimeter field of view. It means now we can take large organs like whole human brain, take say like corona size of both hemispheres and stick it right onto the chip, look at everything with that kind of resolution unbiasedly. So those are the key advantages, at least from those metrics that put us far ahead of the competition. And we continue to make improvements for this technology. Currently, we are working with fresh frozen samples as well as PFA samples, but we will also allow for FFPE samples by end of this year as well. So let me quickly bring you on the hood how biochemistry works. As Wenqi mentioned, MGI had this cool DMB sequencing technology, and they managed, to achieve massively parallel sequencing of short reads by using silicon chips up to 13 by 13 centimeters with a resolution of center to center distance 500 micron between all the different DMBs in a perfectly graded array. And in the last 3 years, we took tremendous efforts to apply this to spatial genomics and made it work. Now we can take a tissue slice, load, fix, and permeabilize on this sequencing chip and make all the DMB as the capture probe to capture the gene expression information. As Wenqi mentioned, each DMB spot has 300 to 500 copies or 300 to 500 probes that can capture the RNA information. So this is the basis how we are able to achieve large field of view, yet still able to keep such fine resolution. As you can see, right over here, the way we enable a spatial analysis is that each DMB will generate, a unique coordinate ID of 25 base pair. So we first sequence all the, UID and get a separate file that has complete X and Y axis information, which containing, the coordinate ID information. And when we actually do the capture, each one of the capture grids is also linked to a unique molecular barcode together with this CID. In this way, in silicon, we are able to see all the captured reads according to its X and Y axis, and was also able to determine if the captured transcripts are from a separate molecule or it is just a duplicate. And another important piece of information here is that the current version of our kit has the poly-T capture probe, and we are also expanding this to capture total RNA instead of just mRNA. And this is a workflow how the STEOSIC experiment will be conducted. We have a very clear and detailed protocol to do the tissue preparation, and our collaborators will follow the SOP and embed the samples either in OCT if it's a fresh frozen, or cross-link with using PFA protocol. And then we'll also be able to expand sample types in the near future. And then the tissue block will be sent over to us, and after QC, we'll do the permeabilization experiment to figure out the best permeabilization condition for each tissue block. Different permeabilization parameters are required to get the optimal STEOSIC library preparation results. And this is done on a separate chip, which we call FC chip. F stands for fluorescence. And this chip is purely for permeabilization optimization experiment. So after figuring out the best permeabilization condition, we proceed with in situ MRI capture on a real STEOSIC chip. The basic STEOSIC chip is called S1 chip, which has a capture area of 1 centimeter by 1 centimeter. As I mentioned previously, it can go up to 13 by 13. We slice the tissue with a recommended thickness of 5 to 20 micron. Generally, 10 micron works the best. We also do a single-strand DNA staining on the same tissue, on the same captured tissue, which will help us with later on cell segmentation. And then after reverse transcription on chip, we connect all the cDNA here from the chip and elute and connect into an epitope tube and proceed with library prep as well as sequencing. Our library is only compatible with MGI sequencer. And then as I just mentioned, currently we are capture all mRNAs. Moving forward, we'll have probes that capture all RNAs. And then after it's reconstructed in silicon, we can visualize using a cloud-based platform called SteroMap. This is just a pic of how what it looks like on the a platform. This is a in-house developed software that has a lot of tool for visualize the result in space. This is the olfactory region of the mouse spring, and you can see cell clusters in this image. Okay. So we have had a few rounds of publications around this STEOSIC technology over the last year. Possibly you have read some of the papers, and two of them become Cell and Science cover paper. The first one is talking about the mouse organogenesis, which is also the technology paper. And then last, I think 2 weeks ago, we have this axolotl regeneration paper published on Science, which I can quickly go over in my next few slides. And there are also papers coming out on the bioRxiv on I would say weekly basis, because we have been applying this technology on different applications for 2 to 3 years now. All right, this is our first technology paper, and it talks about stereoseq use in mouse organogenesis, the process of which is relatively well understood from imaging standpoint of view. People know what's going on, broadly speaking, but it has been poorly understood exactly where the molecular transcriptome is triggered, development trajectory of different cells, and what determines cell fate changes, like those in-depth biologic questions. So on the left-hand corner, you see all the different cell types we were able to analyze using stereoseq data, combining with single cell sequencing results, projecting them into stereoseq map. The stereoseq is able to provide large field of view in an unbiased way, but currently we do like none of the spatial genomics tools for now can offer the same capture efficiency as single cell sequencing. So for this study, we were able to capture 500 to 600 genes per cells with UMI count of 1,300 to 1,400 per cells. It's when you look at the single cell level, it's not whole transcriptome, but it doesn't mean we are only working with those 500 or 600 genes for the entire thing. It just means that when you bring the bean size up, we can, or when you look at larger bean size to multiple cells or 100 cells, we can get the whole transcriptome level back. And so in this study, in order to improve the understanding of different cell types in space, we combine the stereoseq data with single cell data and to be able to impute and map them to where they are located. Of course, certain limitation still exists, which is that very like rare cell types with rare expression sometimes might be hard to be imputed. But so far, at least for brain tissue, we found it is particularly, this brain tissue is particularly suitable for stereoseq. And by doing so across different time points, including 14 middle and late gestation mouse embryos, we were able to recapitulate the development process in pseudo time. And some of the analysis challenges that we have to take into spatial constrained clustering. We did all the analysis using unsupervised spatial constrained clustering to first identify, this is a better example, to first better identify a broader region of where cell types are localized. Then we did subregions and get even more cell types like this within the subregions. And another thing we were able to do successfully with this paper is that in addition to the bean size, so which means you just look at the square size, do the analysis, and we were able to utilize this nuclei staining picture and imputate, like have the boundary of individual cells and superimpose with the green signal are the RNA trends we captured. So superimpose those images together. As you can see, this image suggests that our capture, is indeed at single cell level. We can see a lot of different RNA transcripts within a single nuclei. And then we can use this piece of information to do the segmentation based on cell size instead of just the square size. And now we can see all the cell types in an even higher resolution manner. So yeah, and then there's some data from there are a lot of interesting findings from this paper, and I encourage everyone to read it. And we also have all the stereoseq data online. The link, the database have all the, we uploaded all the STOmics data to this MOSTA database, and the link, I can post the link in the chat box after my talk today, or you can find it from this paper. And this is another cool paper I was just mentioning on Science cover. This is by applying STOmics, we reconstructed the axolotl regeneration with gene expression profiling at single cell resolution. Again, this is an image showing that our technology truly can achieve single cell resolution. The white blob thing is the single strand DNA dye enabling the nuclei. And then each black spots are the physical existence of the DNB spot, which has 300 to 400 copies of capture probe. So with this resolution, and we can see we did a time course study of 60 days after the damage, and you can see how nicely those different cell types grow back, and they find one specific type of glial cells stays active during this process. So this is a paper on Drosophila, has been published on development cell back in May. It's just using the similar approach that I just went over, but we were able to do a 3D model from different stages for Drosophila embryos as well as larvae. And again, this is just to most of the data is just a validation. But there is also a lot of data in the database. We also uploaded to our the STOmics database, and you can find the sequencing results from or raw data from those database and play around with the data yourself. Zebrafish. So we also demonstrated its the application on Zebrafish as well as on the this is a plant, and plant has a very unique advantage that they have cell walls, which is a problem for single cell sequencing, but this is actually a huge advantage for STOmics because you can easily do cell segmentation this way. And this is first spatial transcriptomic characterization in Arabidopsis leaves. Of course, this can be used to give us some insights in the tumor microenvironment and exactly how normal cells, tumor cells, and immune cells interact in space. This has far-reaching application in the future for developing new digital pathology platforms, and because we can definitely provide much more information than existing pathology current method, than with just using H&E staining. So I'm just gonna quickly, go over how what we offer and how we can collaborate in the United States. So basically, we have set up a lab in San Jose, which will, provide end-to-end solution to our US collaborators. What you will do is just to prepare the tissue blocks using the detailed protocol we sent to you, and then you can send the tissue block to the our San Jose lab. And we will do the in situ RNA capture as well as library construction sequencing and return you the the data either through hard drive or we are also setting up a cloud-based system for the data delivery as well. And you will have the access to our stereo map, the online visualization tool I just described. And you will receive a summary analysis report, which include like the fluorescence image I was mentioning, and some basic analysis results, including genome alignment and quantification of gene expression or cluster, typical, cluster analysis using UMAP, and some pseudo-time analysis if there's any, and annotation interaction of those cell cluster, and some basic functional enrichment analysis. You will get FASTQ raw data back, as well as some of the files that generated along the analysis pipeline, including BAM, NANSO, and some cluster, files. And there are some other, information you might get for human and mouse only. And we'd like to, work with you for technology development, data sharing, resource sharing, as well as, we can also co-publish paper together. There are many ways we can, collaborate. If you are interested, feel free to, contact me and or, my other colleagues, and so that we can, work out a path that we can, you can have access to our STEOmics technology. Okay, I think that's the, end of my presentation. And thanks everyone. And we'd like to take questions.
If you look in the chat, it looks like we have a handful of questions. If you can see those from the top, the first one seems to be, what is CID stand for?
Coordinate ID, which will provide the axis, y-axis information from the chip. Let me go back to the this slide. Yeah, so each unique DMB spot will have a unique 25 base pair barcode, and that barcode will tell you the location of the the DMB, and which will be used for the later spatial genomics analysis to assign the RNA transcripts to individual DMB spots.
And I think you can see the next question from Anoja is when the
yeah, I'm trying to get back to, maybe I have to stop sharing so that I can see the question. Let me see if I can see the question. Oh, I do. All right. What's the, when will the commercial product be available for STEOmics? And do we have to purchase MGI sequencer for process? Good question. So we are in the middle of commercializing our product. It's not, it's possibly, for next year, maybe around Q2. And yes, as I mentioned, the STEOmics currently, the design of the probe is only compatible with MGI sequencer. So we have to use MGI sequencer for processing the data. Would there be a future option for local in-house STEOmics end-to-end performed in a core facility? Of course, yes. We have different business model in mind, and that's definitely one of them.
Well, terrific. Are there other questions? Everyone should feel free to post them in the chat if you'd like. We'll just give another second. But
Sure.
Wenxi and Yuan, thank you both very much. We appreciated that content. We'll remind everyone that next week's session will be presented by Oxford Nanopore Technologies. Once again, at the same time, same day on Thursday at 2 p.m. Eastern. We look forward to seeing everyone then. As a final reminder, as I said, the recording of today's session will be sent to everyone and also posted on the workshops and webinars page of the ABRF website. Anoja, thank you. Thank you to our colleagues at MGI, and I hope everyone has a terrific rest of their day. Have a great day, everyone. Bye now.
Thanks, Ken. Thanks, everyone.